resource. Okay. For one thing, variants of unknown significance were more frequent in these patients, as one might expect, in underrepresented and diverse international populations. In discussion of some of the cases, it was clear that microarrays were important to diagnosis, and we were able to add microarrays in selected situations. Everyone agreed that there was very strong educational and clinical value to the pre and post sequencing case conferences that we had. Of course, as shown, these exomes led to diagnosis in some, but not all, unsolved cases. We've talked a lot about how the field is moving more now towards reflex or even first line whole genome sequencing, also potentially RNA-seq, long-read technologies, and something we might talk about as we implement a larger project, that maybe there could be a research piece to this in which we can expand beyond exomes. Fabric software was used by a few sites, and the main limitation there is that some of the sites just didn't have the local expertise or the personnel time to be able to delve into the exome sequence data, so something we can examine and think about a bit further as we think about the expansion. Further workup, as I mentioned, was suggested in a number of equivocal cases. And maybe one of the most important things that we've learned is that we had 6 sites, and each site was its own story in terms of the logistics of local implementation. And we needed to provide each of the sites autonomy to develop their own workflows, their own clinical practices, to decide how to allocate the limited resources that we had available to them. And I think that that's going to be an important theme as we go from 6 sites to potentially 20 plus. I think that may be, yeah. So in terms of ongoing plans, we do have several plans for publications, and I won't go into all that in detail because I want to have a lot of discussion to follow. And we're planning on submitting pathogenic variants to ClinVar, and also some of the novel population-specific variants to the DB SNP. And of course, we want to scale up. So I don't know, we showed this slide at least 3 or 4 years ago about our dream for what a G2MC rare undiagnosed disease project would look like. There were 20 plus clinical sites that really very interested in participating. And the structure here, shows those clinical sites throughout the world. We're not the only people working in this arena, and there are other terrific organizations that are also working hard in the rare undiagnosed disease space. We want to collaborate and work with them, not compete with them. And we've been in touch with a number of them over the years, and need to re-engage them more as we go forward during this expansion. We think there'll have to be some kind of a coordinating role of a coordinating center of sorts, and G2MC and potentially the University of Maryland might be those sites, but all up for discussion. And we need a sequencing partner. And again, up for discussion as to what model we use, whether it be a research model versus a clinical model. We saw that we went more of a clinical model for the pilot study, sequencing being done in a clinical lab. But there's also the possibility as we scale up, if there are going to be tens of thousands of exomes to be sequenced, potentially a research venue followed by clinical validation that may be more efficient. And the work that I do at the Regeneron Genetics Center might be an opportunity, but all options are open for a sequencing part. So in terms of the scale up and open for discussion, please go ahead. Yes, so we'll talk about that, but one model, I don't have me from Maryland who joined by Zoom, unfortunately, is to use a central IRB. and then the consents would all be formatted more or less the same in multiple languages. They would probably likely need to undergo secondary review by a local IRB, ultimately for approvals. But that could very well streamline a lot of the ethical and IRB and ethics committee issues that might occur, especially if a place like the Regeneron Genetics Center, the place where the samples landed. So needs to be worked out, but that's one thought. Okay, so in terms of scale up, we need to talk about research versus clinical models, both on the table, and neither mutually exclusive of one another. There might be some sites that might function better, more easily in a clinical model, and others in a research model. So we'll talk about that. To George's point, the whole IRB issues, central IRB versus something different. This is done in a research center. In all likelihood, there needs to be clinical validation of that variation before results are returned. But there are local regulations and laws in all different countries, and that may differ, and we need to discuss how that would work. The clinical model, as you mentioned, likely we can get IRB, non-human subjects research designation, and that journey may be a little bit easier, but then funding sources may be a little bit more difficult to come by. Data sharing and publications may also be a little bit more limiting in a clinical model. And as I said, the options are not mutually exclusive. So that's my last slide. And what I'd love to do is get input from the group about, first of all, any comments about the pilot study. We have many of the clinical site PIs here comment further. And then let's launch into what an expansion might look like.
Not a comment, but a question, and also reflecting on Gabriela's presentation. What do you think is the best way of analyzing the sequencing results? I understand that in your model, what the clinicians review on the local sites are just the variants. There may be a few diagnoses lost in the processing between, the crude sequencing data and then the processed variant coding file. What is the best way? What would you recommend for an implementation on a large scale? What would be the best choice not to lose a few diagnoses or to come back to the diagnosis if you miss them through these processings? Is it possible to, what did you do and what do you recommend?
May I respond that from coming from a site? Because I think it all depends, Mark, on whether you have the local capacity to analyze data. So, for example, we had sites which used the report which came from Centrogene directly. But we also enabled the local sites to obtain the raw data and to analyze it using the software provided by Fabric. That option also we provided, and not every site used that. Thirdly, we had my one in Colombo where we had our analyze. So in this model, all three options were available. But it all depended on whether the site data analytic capability. Even when provided the Fabric software, some sites they only had the clinicians that maybe they didn't find. Was spot on, but this project so there are
Non-human. I'll check. I don't think it was exempt. It was non-human. I'll check.
think you're spot on again. Like we can definitely use that, and that model of case discussions, I think, was very useful for the young investigators in the site, and we'll listen to them in a minute. So most certainly, and then of course our next step of publishing this as a series, and so let me invite Claudia, yeah, just to comment on that. Yeah. Oh, we got it very fast.
Within
yeah,
a month, right?
Within a month. Yeah, we had within a month. Yeah. Claudia, could you comment on our plans for publishing? And Teji, I saw you coming in, Teji, and I think from the young investigators' point of view, they're all asking about publication fees, whether RTW has, yes. And in the current publications that are going in, we have to pay for the publication fee, and that RTW has approved. Okay. Yeah. Okay. Fine. Okay. Over to Claudia.
Yeah, so as Alan mentioned, so one of the things we looked into as part of the group was starting a special issue on research and diagnosis of rare diseases in low and middle income countries. This is a special issue in Frontiers in Genetics. Rajira, myself, Atanu, and Fernando Casals are editors for the special issue. And it is not just open for the cases of this pilot project, but we opened it up for, in general, reports on rare diseases in LMICs. Because as we know, sometimes, I mean, in higher income countries, they have large numbers, big projects like Decipher and so, and sometimes in LMICs, we have a few cases and they don't get the as much spotlight as other bigger projects. So that was part of the idea. So submission is open till mid-November. And yeah, so we'll be reporting everything that Alan just presented on the pilot as one of the papers in this issue. But it's also open for the case reports and again, just general research or insights into how to implement genomic diagnostics in LMICs.
So we've got till November. Two questions, hands up. Gabriela.
So I have two questions about the touching the capacity building and the scale up. So one of the issues you mentioned was the difference in capacities for bioinformatic analysis. And also think about how could we link up with other working groups from G2MC. Can there be like a teaching module for within, like, for example, the education working group for analysis and interpretation, so we really build up at least those capacities in each of the participating groups? Do you think that that would be feasible? And the second question is also how do you see, I mean, I know that Centogene and Fabric are really like well reputed and very solid industries in the field, but can we also perhaps take the risk of looking at capacities in LMICs and probably try to do at least part of this in other places? I mean, I'm just thinking of Latin America. Well, Claudia just mentioned equipment that's being underused. And we know, like, for example, Brazil and Argentina are developing models. So we could also help these LMICs develop and grow their own initiatives along with this project. Might be a little bit riskier, but I think it would be really powerful if we try to do something along those lines.
Thank you for that, Gabriela. I think I will let Rich answer the education question, but I'll answer the second one. Yes, I think what we've been, I think myself, Alan, we've been brainstorming about this. And so what we find is that every site does not have every capability. So we were even also thinking of requesting the sites that are coming in now to look for other capabilities in your countries, and share, maybe bring together bioinformaticians, scientists, and clinicians. So work as a, even these sites can be a collaborative network within the country, rather than just one person collaborating with G2MC, so that there is capacity that develops in that country. So that was one of the thinkings that was going on in our minds, because last time when we said, okay, identify an education lead, identify an informatics lead, some of the sites were unable to do that, because simply they didn't have those numbers of people. So going forward, I think in the scale up, Alan, as we were discussing, we need to kind of refine our, ask from the sites, and that would mean, identify your educational needs, identify your bioinformatics, whether collaborating network in your country, so that this becomes a, global enterprise that really catches on. So I think that's the way to go for that. So, Rich, any suggestions on the education? Yeah.
Doing that stuff.
Yeah.
Or, because, it's a whole process that we really. It's funny, you're doing.
Yeah.
It's a great.
So, so we had clinical case conferences where there was lots of participation, and I think people took back the learning from those clinical case discussions back into their practice. But in terms of the bioinformatics analysis, we did have, training sessions as such, or introduction sessions to software. But I think it didn't, at the sites did not have people implementing it yet. So, Claudia, you have. Yes.
I have a comment that I would like to build from what we have discussed this morning, which is related to the cost and the benefit from doing NGS. We have to admit that NGS costs are getting lower and lower, and even with these capabilities, IT capabilities, artificial intelligence, I don't think that software are really a limiting point in this process. But with this, we have also to admit that our understanding, our analysis of WES, like you have shown here, doesn't exceed 30%. Part of it is related to undiagnosed phenotype, but What about the 50% which are phenotypes that are clinically well diagnosed, well defined? So I'm wondering here whether we have to switch or to change our analysis. We usually start from the clinics and then try to identify the genetic etiology of this phenotype. Why we don't go the other way? And I think having multidisciplinary staff, starting from the bioinformatician, from the bioinformatician, targeting the most important variants, and then trying to identify the clinical features that can explain these variants can be a possible or an alternative to increase this gap or to reduce this gap.
Thank you for that suggestion. I think that's, you know, as we go on, it maybe we could put together a group which can work on data to see why, you know, to increase the diagnostic yield with all this. And of course, we've been discussing the fact that some of these, you know, populations that we are working on are underrepresented in the databases, and we also need to have some data on the population, frequencies of variants and so on. So that's another, you know, big thing that we need to really work on. Bruce, you had something.
I'm wondering, first of all, if you take all the people who have been diagnosed collectively, and it would include not just, excuse me, from this project, but all the others that you're referencing here, begin to synthesize what are the clues that predict the ability to make a diagnosis, to help refine the kind of selectivity of individuals that are subject to sequencing. And then on the other side, the undiagnosed group. I suspect that some of them will still have genetic causes that as technologies improve, they'll succumb to diagnostics. But it raises the interesting question, maybe there are other things going on entirely that are not genetic in the way we usually think of genetic, either multiple genes involved, if genes are involved at all, or epigenetic changes or whatever. But that becomes a really interesting population to begin to dissect out what some of the etiological factors are that may actually reveal mechanisms that we're fundamentally unfamiliar with right now.
Absolutely, that's I think something really valuable we can bring into that bring onto the table. Bruce, we'll need to give some thoughts on how we. Jorgen, and then Claudia, sorry.
Just two quick comments. One is, so based on the experience from the pilot, I thought there were some things that were really useful. So the ability to send blood spots to Centogene rather than DNA facilitated a lot of the process. But then sometimes we had issues with getting the data from Centogene, the raw data, or when we had microarrays run, we didn't get that data. So then there's a little bit of a trade-off. So just as a comment for reflection into the scale up. And then the other comment is about what Gabriela mentioned. I think that considering partnering with local infrastructure, whether that's bioinformaticians, but also labs. So one of the main issues that we have in Latin America, in Mexico, is that there are very few labs, but there are some that do genomic sequencing, and they struggle because, again, they cannot compete with price for sending samples to Centogene or to Invite. But I think as part of that capacity building, if we sort of determine certain parameters, right? Like if we are all going to do clinical exome sequencing, these are the sort of requirements. And if a lab in the country or the region, because we could also see it as a regional organization as well, fulfill those requirements, I think it's worth, and at a reasonable within range price, I think it's worth engaging with that local capacity to build that up and create those markets in those regions.
Yeah, absolutely. We need to work with the technology providers on the model.
So I always sit in the back and think crazy stuff. And so let me try and sort of throw out a few interesting ideas. So Bruce mentioned something that so first of all, I think doing whole genome sequencing is a no-brainer. Ought to be done. But Bruce threw out an interesting idea, which has been mulling around in my head for a long time now. Think about rare diseases as being monogenic. Maybe they're also rare because they're multigenic and multi-allelic. So once again, doing whole genome sequencing might be important. Now, you have a list of countries up there, and I think especially when you're dealing with Latin American countries, and the one that I know best is Ecuador, turns out we know very little about the populations that live there. Of course, in Ecuador, you have the Spanish conquistadores, they brought their genes, but in Ecuador, there are at least 4 indigenous languages. So if you assume that language represents population groups, there are at least 4 indigenous population groups, or probably more. And African slaves were brought in to harvest sugarcane. And I was at a human genome meeting in Brazil. Turns out the populations that have been studied from Africa do not represent the enslaved populations that were brought to Latin America. So there's an additional issue with Afro-Ecuadorians that probably is similar to the one in Brazil, but nobody really knows. And then somebody brought up, I think it might have been you, multidisciplinary approaches. I think throwing the kitchen sink at it, so all the biochemical testing that's done, but also multi-omic approaches are going to turn out to be important. And again, sort of thinking crazy things in the back, we know very little about protein-protein interactions, and some of the variants are likely to affect protein-protein interactions and cause a phenotype. And I don't think we have any good methods to look at that. But it's a basic science challenge, and I hope most of us here are also interested in basic science. So,
thank you, Jon. I mean, we agree with everything that you said, and I think we need to find a way of dealing with it. Let me, ask Rupesh first. Rupesh is from Nepal, and he's a one-man.
That's why I just take the mic. I'd like to congratulate both of you for this project. And I have a question. Like last time, as Professor Bajira has just said, I am the only one person trying to work out in the population, and I was able to send the samples and all. But why don't, like the bioinformatics things is completely computer and AI work. But I believe we have the team of bioinformaticians in some of the sites. As we are from the like low middle income country, we can't expect that every site will have a bioinformatician or a good lab. So there should be some certain things we need to consider, like we send the data to Sri Lanka like last time. So is there a way we can collaborate with, we can say the other sites that if you don't have a bioinformaticians, why don't you work with the other sites and let's make it happen in a good way.
This was what Gabriela was saying, like let's
yes.
have a, I mean, it can be in country or maybe in region.
Yes.
It could be some, yes. So we know, a lot of you may not know, we know sequenced the first Indian genome many years ago. Yeah, over to you.
Yeah, so trained as a clinician and who took up computational biology later, what I can say for a matter of fact is that you don't need a bioinformatician to analyze genomes, right? I've been trained 20 years, 25 years ago, and I've seen how CTs and MRIs have evolved from a data-intensive physical construct to something that clinicians can read day in, day out, right? I mean, I would rather trust my neurologist to read the CT or MRI rather than a radiologist to read that. I mean, so genomics is also reaching to that point in time when you don't require a bioinformatician to do that. I mean, having said that, I'm a bioinformatician myself. I've trained enough number of clinicians with the necessary skill sets to enable them to read the genomic data. And I think that is the real power of genomics. If you don't give that power to a specialist, you are not really going to take out the real value of genomics. Now, there are methods we can train clinicians into how to use genomic data in a day-to-day practice, which doesn't require bioinformatics capacity, because a lot of automated stuff can actually be taken care by computers today. And you just need to give that power in front of a clinician so that they can act upon that information. And to give facts, we actually have specialist clinicians pinpoint stuff that bioinformaticians could not look at, because bioinformaticians, as you know, are generalists. right? They will look from anything from muscular dystrophy to neurodegenerative disease to sarcoidosis to anything. They're experts in none. But specialists look at one specialization, whether it is rheumatology, look at autoinflammatory diseases, they know it inside out, right? So they are better equipped to analyze genomic data in the coming years. Thank you.
Thanks. And yeah, going forward, I think we can tap into Vinod's expertise. Yes, done so much in India. He just left government, so he's free to work for us as of today. So he will, use his tap into him. Rich, yes. Yes. Yeah. What is, yeah. Yeah. Absolutely. Yes. Yeah. Yeah. Absolutely. Yeah. Yeah, I can tell you my own experience in Sri Lanka. I have a sequencer in-house, but then can't sequence because the prices are so, expensive. The supply chain is kind of going off the roof. And we decided what is more important for us is getting it sequenced where it can be done for an affordable price. And let's work with the data and the patients. And so we've now got a very robust bioinformatics pipeline of the clinicians looking at the data, and we make decisions very comfortably, and we have an idea background genetic makeup of the population. So but then of course, as you know, Rich, when you look at the wider population and the wider community and expectation of the people of the country and so on, they'd like to be saying, okay, we are not sending our samples abroad, you are doing it here, and all that nationalistic feelings and all of these things play up. And I think they are everywhere in every country.
With the broader research, because I think I agree with that. So we're reaching, especially for exomes, the point where it's pretty standardized. Clinical laboratories have SOPs for running these tests, and you have software that is really easy to call pathogenic, likely pathogenic, but that's still limited to reported genes and things that are known. So in that sense, I think, yes, either training and empowering clinicians to interpret that, it's useful, but also we need a path for all those 40% or so of cases that won't have a slam dunk finding, right? So, and especially for underrepresented populations, we see it and Gabriela might have also pointed at that. We see a lot of variants of unknown significance, even in known genes, because we just haven't characterized those variants. So I think we also need to establish those paths to send these for research purposes, for reanalyzing the exome on a research basis, for doing reflex whole genomes and functional testing and all the other stuff that it's not needed when you have a really easy clinical finding, but there's still that huge gap on the research side.
Absolutely.
Because we're in a zone, sometimes you do need a radiologist to tell you that a spot is not pathology. We are at a time of maturation of our tool where we have a very strong push to make a diagnosis of undiagnosed cases, and where you have some variants of uncertain significance, which are a gray zone. You have to make a decision between not making a diagnosis that might be or making one that might be false. And my personal point of view is that you're better off without a diagnosis than with a wrong diagnosis, which everyone will believe very strongly because it is inherited and it is high technology. So I think we should still be relatively cautious in the way that we transmit this information to the clinicians who can use it as a black box.
Thank you. Yes.
At a point, I think that's a very valid concern. And in fact, global databases, whether you take ClinVar, are actually rife with false positive variants just because the variants were not evaluated at a population scale in terms of allele frequency. They were considered as pathogenic, and they've been followed as pathogenic over and over again. So, I mean, the papers after papers which have come back saying that these are pathogenic variants, but they're very polymorphic in the population. So I think the population control group that you have thought about is really a valid point to make, because without that, we would just keep on adding this false positive information in literature.
Chris, did you put your hand up? Did you put your hand up? No. Okay, I thought you had something. Yeah, okay, Yasas. So Yasas was our lead investigator in Sri Lanka. If you want.
Yeah, and comment and suggestion about our project. I'm really thankful for the organizers for having this committee. And regarding the cases, I really love the case conferences, especially the pre-case conferences, because we learned a lot from each other sides as well as from the developed nations, and how we can get whether to go for the microarray, when to go for like that. So the one suggestion is to have some sort of, when we are presenting cases, whether we can have subgroups like skeletal subgroup, and because when we are doing a scale-up project, then it would be easier if we can brought up some expert clinicians as well for that. I want to hear the others comments as well. Thank you very much.
That's a great suggestion. I think, yeah, because we had a wide array of cases and we were, lumping everything together and discussing. But as you said, like in a scale up, if we, categorize the groups, that'll definitely, and with the clinicians coming in, yes, that's a great suggestion. Thank you. So Winda, you were in Sri Lanka and in our project, and now you are in UK. What's the difference in your practice there and the practice in Sri Lanka? Is there any learning that we can, bring into our project to enable?
Yeah, so regarding actually, with the data, when we find VUSs, we are in great trouble what to do, what to tell to the patients and all. So actually, from the experience that I've gained from UK for this short period, so what I learned is they are usually, before implementing testing, they have, usually in UK, they do gene panels. So depending on the patient's symptoms, they select which panel they do, but it is, most of them are either whole exome sequencing or genome sequencing. But the clinical evaluation is what is the, they consider as first priority. For example, if I take one example, your autism, if you do genetic testing for a child with autism, they are really cautious to test beyond microarray, because finding a VUS is not fair by the patient. So they're really cautious when they do whole genome sequencing in a child with autism. So if it is really valid, they usually discuss in among the colleagues in the department and take consensus before they proceed with the testing. So So the clinical expertise should come first, and then when you do the testing, the bioinformatician role is really important in variant interpretations and stuff. And so regarding this project, I think those are the undiagnosed cases. I think it's better in the scale-up project if we can reevaluate them clinically and then bioinformatically. So we might be able to learn more lessons, I guess.
So what you're saying is we should be careful in the phenotypes that we include in our scale-up.
Yes.
Which ones are we really going to?
Yeah. So if we can like reanalyze the once we have not found an answer, I think as like Yasas mentioned, among clinicians to reevaluate where we have missed it, why we haven't find an answer for that. Starting from clinical, then whether to reanalyze data or not, taking decisions like that would be of good exercise for every one of us.
Thank you, Jorgen.
Overloaded medical doctors. So once we come up with diagnosis, and I don't know how this applies to other LMICs, but if it were like in Ecuador, the healthcare system tends to be overloaded like everywhere else. So the medical doctors may not give the best possible genetic counseling. What I often think about is the indigenous woman who lives on a subsistence farm in the Andes. We've got to relate this relatively complex information that we're struggling with to actually figuring out a diagnosis to what it means to her life and her family. This is not an immediate problem, but this is a problem that at some point I think will come up in most countries and certainly in LMIC countries that have very diverse populations.
Thank you. I just like to switch gear and bring in another aspect that was mentioned in the previous session also, like piggy banking pharmacogenomics on the rare disorders project. And this was something in some earlier discussions that George was pointing out to me. George, any views on piggy banking pharmacogenomics on the rare disorders?
Well, I think it's quite an opportunity to have information from those particular cohorts because we can easily extract this information from the exomes or the genomes. Well, talking about exomes, so it has to be exomes this case and perhaps impute, get what is needed. I'm not sure if having such a small number of the anticipated samples to give a picture, because normally a population pharmacogenomics approach would entail to have a much larger sample pool. But I guess we could go along with what we have at hand and perhaps try to coordinate with the existing databases that we could extract more information. But we're clearly moving towards the population pharmacogenomics concept, which is moving from, especially also along with the pan-genome concept, right? So moving along that direction and considering the fact that pharmacogenomics is the low-hanging fruit, then perhaps it's worth focusing on that as well. Well, I think for such an answer, one would need to do a population, a power calculation first. Well, in my view, it's better to start even if we have 18, 20, 50 genomes, sequences, let's say exomes, start categorizing in a basic database format, and then provide the opportunity to expand as much as information, more information becomes available. That's what I would think. So I wouldn't wait until we get, let's say, over 200 or 300 samples, because then it would be a hassle to create such a resource. So why not starting small? And once we get more information, we build and we extract automatically from that. That's actually what we've done in our 2014 paper.
Thank you, George. Any others? Views, suggestions? Yeah, yes,
Regarding the Fabric software, what is the role of Fabric software in the scale-up project? And are we going to let the individual sites to decide on when to use Fabric software in the scale-up project as well? What are your thoughts on that?
We haven't made a decision. That's why we are. Yes. Yes. Utilize it. Yes. Okay. Yeah. Yeah, access software and use it. Yeah. Yeah. Yeah, Tej. Yeah. So I think sites have to respond to Teji's email, whether they want. I just want to ask MK, the experience of the Malaysian site.
Thank you, sure. Yes, we basically the site has worked quite well within our own locality, but as we expand it into more areas, particularly in many parts of the country, I think we're going to have some issues with the counseling, the preparation of samples, the logistics, and so on. And this would be an issue that we need to look into, because no point getting poor quality samples and having to get patients to come back again and again. So we probably need to look at various other ways of collecting samples, perhaps like dried blood spots and other ways of getting it done. I think there are issues that as we expand beyond, teaching facilities and universities, I think there would be issues.
All right, thank you. Any other comments? I was wondering, Devendra is also here. Devendra, you were having a Decipher India project, I think. Any experience from that you can share?
Yeah, thank you, Chair. I missed some of the session. I came a bit late. I'm sorry. Yeah, I think any effort in the low and middle income countries for rare diseases or even common diseases is welcome. There are logistics, as you know. There are some legal barriers imposed by the country, relevant country like India, and the lot of main challenge in a large country like India is the population heterogeneity. As you know, there are 4,000 distinct population groups, which we know has been involved in genome sequencing, as you know, I think more than I do, because I'm Indian origin but based in the UK. The last many years, we developed a program, which Vajir also helping me. It's called UK-India Genomic Medicine Alliance. So there are many projects. One project the main project is the birth defects and rare undiagnosed diseases. What we started, as you know, the Sanger project called deciphering the developmental disorders called D. Started by Professor Helen First in the Sanger Institute. It's a very prime project. It has got its very own specific database, which is just like ClinGen and ClinVar, but very distinct. One of the most important elements of this project, the Decipher project, is a deep phenotyping, which has been mentioned recently in this discussion just now. For example, if you have got a variant of unknown significance, so most people who are not aware, and you look at it, it's of no significance. They either interpret normal, it goes into records. patient is very happy, the family is very happy, it is normal, which is of course mistake, which could be revised or upgraded. The main thing about the decipher is that it's a continuous process. So that is every week it is upgraded. What we have done, we have created a project called Decipher India Consortium. It's a consortium. It's not like that there is a project behind all. It's a continuous process. So about 12 or 14 centers included. There are, some of them are government institutions, some are private, all included. They are welcome. What we are doing, I think every center has got either individual registration or the institutional registration to go put their data into Decipher. And once it is there, the variant unknown significance with the phenotyping, it gives opportunity for the person, for the clinician, I mean, to go and look at their own patient, go back and see what we have missed on the list. Suppose there are 2 cases reported with the same variant, or maybe in one case pathogenic, what they can see, they can compare the patient. So the idea is to go back, whether you go back yourself, or even now, the trend, as you know, is called human phenotype ontology based phenotyping. I think that's what we are doing, and we are building up slowly in India. And in fact, I mean, I would say it's South Asia, but Geneva also volunteered in that. So I think it's a very open project and based on the Decipher experiences Sanger. Thank you.
Sanger Center. So it's basically clinician support in terms of phenotype. Thank you. Any other comments, suggestions? So one of the things that we didn't discuss was the regulatory aspects. So we've been leaving it to the sites to decide how they overcome their own country's regulatory aspects, and also whether they are able to take part in the international. Pretty much even the back responsibility to the site, because cannot really coordinate across the world and in every jurisdictions. Yeah. Yeah, we need to think about it. Yeah, no, we don't have an answer to that at the moment. We need to think about it and give some thought to it. Very difficult. Yeah. No, no. Yeah. Oh yes, most no problem. It can be done. Yes. Yeah. Yeah, we'd love to. I mean, we haven't closed any for any new sites. Yes, most certainly. Okay then. So thank you very much. It was great. Thank you. A little announcement.
Really quick from the communications team, if you have any kind of social media, whether you're using LinkedIn or Twitter, etc. First off, we love the promotion that you guys are doing on your end, and we'd love to keep encouraging that. The only thing we ask is that you add hashtag G2MC Geneva at the end of your post to keep everything sort of together. It helps our engagement. It'll help your engagement as well. Otherwise, keep posting. We really appreciate it.